everybody, and welcome to part 4 of your biology unit. In this unit we're going to be looking at enzymes and digestion. As this is quite a big unit, I have split the syllabus into 2 different sections. So we're doing part 1, which includes 4.1, part of 4.2. If you look at your syllabus, you'll see there's more, and 4.3. Part 2 of this will be looking at the rest of 4.2. So in 4.1 we're looking at enzymes. What are they? Biological catalysts, what does that mean? And the specific enzymes, including amylase, lipase, protease. I'm looking at the lock and key model to illustrate a thing called substrate specificity. We're also going to be looking at inhibitors. They fit nicely into this, and these are molecules that fit into the active site, means that nothing else can work. And the third one is to demonstrate your knowledge and understanding that in food digestion, enzymes are needed to break down large insoluble molecules. Now these molecules we've just done in the nutrition unit, and we're breaking those large insoluble molecules down into ones that are small enough and soluble enough that can be absorbed into the bloodstream, and that they have commercial and economic uses, including biological washing powders. Now actually we're going to be covering that in part 2, that last sentence on the commercial and economic uses. So the first thing we're going to look at is the function of the digestive system. What does it do? There's a couple of different things that it does, but we're going to look at 2 specific functions. One is digestion, and that is the breaking of large insoluble food molecules into small soluble molecules that can be absorbed. That's your definition that you need to learn. And absorption then is the process in which small soluble food molecules are transferred from the gut into the blood system. Now the ileum there, the small intestine, is an incredibly long piece of gut, and it is twisted and turned to fit into quite a small area. Now running right beside that are a huge capillary network of bloods, because we eat the food, it goes in our mouth, it goes down the esophagus, you can see the picture of it there, it goes down the esophagus, goes into the stomach where it hits the acid, and it's getting broken up as it goes down there, and it then goes into the small intestine where digestion finishes and absorption really happens, and then it goes into the large intestine. You can see there that brown kind of larger squiggly thing there, and that's mainly to do with absorbing water back off the food that is in there to keep the water balance in the body right, and then digestion it out of the body. But you do need to know these 2 definitions. You need to be able to recite those and know those by heart. So, as mentioned in the previous slide, the function of the digestive system is to break down these large insoluble molecules. So what do we mean by that? Now in the previous section we looked at nutrients, and we looked at the idea that proteins are made up of a very long polypeptide chain that has single amino acids. These are all bound together. Lipids there, or fats, are made from glycerol with 3 fatty acid chains on them. We spoke about the length of them and how kinked they are. And we have a carbohydrate chain. That's a simple one there. You also have starch and glycogen and all of these different complex carbohydrates. So that's a simple carbohydrate chain there. And you also have complex carbohydrates, but they're essentially the same thing, a very long chain of sugar molecules. Now, what those 3 molecules have in common is that they're too big to get into the blood, and they're insoluble. That means they don't dissolve. Now, to get into the blood system, you're talking they have to be digested down, right down to the individual molecules to be small enough to get in there. If you imagine the piece of meat that you've just eaten and for your sandwich or whatever it was, if you think about the carbohydrates in the bread and the piece of meat inside it, they're too big to dissolve into blood, so they have to be broken down and broken down and broken down until they can be absorbed then into the blood. For digestion, we're breaking down the food molecules that we have into smaller molecules. So that is, you take the large insoluble molecule and you break it down. Now I've represented this by a pair of scissors, but those bonds that are holding the different molecules together, be it the amino acids, be it the sugar molecules, be it the fatty acids and the glycerols, those bonds are broken down in digestion, and then the smaller units that are left, be it the sugars, be it the amino acids, be it the fatty acids, they're small enough and soluble and they can be absorbed into the blood. The molecule we're going to look at here are your carbohydrates. Now you've got your simple chain of carbohydrates up the top here. Each sugar unit is represented there by a yellowy circle, and the blue line is the bonds, the hydrogen bonds holding those together. So the large carbohydrate molecule cannot be absorbed. It's too big to get into that blood vessel to be able to be dissolved into the plasma. So by cutting the bonds, breaking those bonds up that are holding the sugar units together, those individual sugar units are small enough and soluble to get into the blood vessel. We need to be able to do that because obviously we need the sugar for respiration. We carry that, we need it in every single cell of our body. We carry it around our body in our blood vessels. So it needs to be small enough to be dissolved into the plasma. So this one is the proteins, and again we recognize the picture from the last section on nutrition. So we have our protein chain, our polypeptide chain, and it is made up of different amino acids. Now each one of these single amino acids have different functions, different jobs, but the large polypeptide chain, the large protein chain itself, cannot be absorbed. It's too big to get in. Again, it has to be cut up, it has to be broken down into the individual amino acids, and those individual amino acids are small enough and are soluble, and they can be transferred into the blood vessel from the gut, and that then can be transported around the body. The final molecule we're looking at here are the lipids, or the fats, and again, if you remember that they're made up of a glycerol unit that has 3 ports as such for fatty acids. Now the hydrogen bonds, again, they're represented there by a yellow line. They are cut, they're broken, those bonds are broken, and it separates into a glycerol molecule and 3 fatty acid chains. And that glycerol molecule and those fatty acid chains are small enough and soluble enough to be dissolved across the walls of the blood vessel and to be dissolved into the blood plasma and carried away. So whilst in this section we are going to look particularly at enzymes, there are 2 ways of digesting your food. The first one is mechanical digestion. Think about eating your sandwich. The first thing you do is you take your sandwich, put it into your mouth, and your teeth break off a smaller piece. So that's digesting, that's breaking that larger food unit that you have, breaking it up into small bits. You then put that into your mouth, and very quickly it doesn't resemble what you put in because your teeth pulverize it. They break it down into smaller and smaller pieces. Now digestion does start in your mouth, and in this unit we have concentrated on it in the small intestine, but there is amylase enzyme in the saliva of your mouth, and it does start to break down the starch in your mouth. Now it then goes into the stomach, and the stomach is a big sack of pure acid. It's pH1 acid. It's incredibly strong acid. And once you put the food down in there, it does what you would imagine something will do when you put it into pure acid. It starts to kind of disintegrate into nothing. And what's happening there is the food is being broken down and broken down and broken down into its individual parts. Now there are enzymes that are able to survive in that pH1, the protease enzymes. They can survive in that very toxic environment that is the stomach, that very, very acidic environment. But the protease enzymes can survive in there, and they actually do very well. So the digestion of your stomach, of your protein, really starts to happen in the stomach there. Now, the second one then is your chemical digestion, and this is where your food is broken down by enzymes. Like I said, the majority of this happens at the top of the small intestine, but amylase enzyme is in the saliva, protease enzyme is in the stomach. So the chemical digestion is happening as well as the mechanical digestion. And once the food comes out of the stomach, it doesn't resemble what went in, should we say. It has been broken down quite considerably, and that means that those chemicals can actually get to all the parts of the food to start doing their job and breaking it down. So an enzyme is a protein. I've said this before, that proteins come in lots of different shapes. We've got hemoglobin, enzymes, antibodies, and so forth. So enzyme is a protein that acts as a biological catalyst that speeds up reactions without being used in the reaction itself. Now, another place where you would know a catalyst is in a calculator. if you are doing a math sum, that's obviously not a biological catalyst, but if you're doing a math sum, you could probably do it in your head. You know, if you took a very simple math sum, and you would be able to do it in your head. But if you put it into a calculator, you get there faster. And that is what biological catalysts do. They speed up the reaction at which these things are happening. Now, the enzymes or the molecules will break down, but they wouldn't break down fast enough for us to be able to use them as an energy source, for example, for the carbohydrates or so forth. So we have enzymes that will break them down. Now, again, when you use your calculator, you just press reset and your calculator goes back to the beginning. Enzymes are like that. You use them, the substrate, the carbohydrate or whatever goes in, and the enzyme then is reset. So the enzyme does not get used up by the reaction itself. It's like a calculator, it does the job and then goes back next. So an enzyme is a protein that acts as a biological catalyst that speeds up reactions without being used up in the reaction itself. Now that definition is in red for a reason. Now enzymes can break molecules up, and we're looking at digestion, so we are looking at breaking up molecules, but they can actually as well, and do frequently, build molecules up as well. So they can break them down and build them up as well. So we've got just an example of one here, a large insoluble molecule. The enzymes are represented by the scissors. Right, so they do the job. They go in and they break that bond, and the individual subunits come across. So you've got a large insoluble unit, you've got digestion, your enzymes are going in and acting like a pair of scissors, breaking those bonds, and you've got your small soluble units. So how do the enzymes work? Now you've got your enzyme and you've got your substrate. Now your enzyme is your machine as such, your enzyme. Your substrate is the likes of your carbohydrate, your protein, your fat. Another word for that, collectively all of those things are called substrates. Now there's what's called the lock and key model, and that is because the substrate fits into a very specific part of the enzyme, and it's called the active site. Now I've highlighted here in bright pink, and the enzyme is absolutely specific to the substrate, and we'll see this in the next couple of slides. So you have an enzyme that's going to work with a carbohydrate, and it is shaped, the active site is shaped to fit that carbohydrate, and that's it. It won't work with any protein or fat or anything like that because it's not shaped to fit. That active site is very, very specific to one thing, and we do have a lot of enzymes in our body. Because each one of them is specific, we have a lot of them. So you have the enzyme on the left and the substrate on the right. The substrate fits exactly into the active site. Now, so when these two come together, the substrate will move to come in to the active site. You can see there getting closer and closer and closer. Now once that is in the active site, the enzyme will in this case break that substrate down into two separate molecules. So you can see the substrate is broken into two, and then those two separate molecules move out from the active site. The active site remains unchanged and is ready for the next substrate to move into it. Well, enzyme substrate complex. It's not as fancy as it sounds. What happens is that you've got the substrate and the enzyme, and they fit very tightly together. Now the substrate goes in, there's a very tight fit, like you would be putting a key into a lock. Once that substrate goes in, it's broken down into the components. For example, a carbohydrate is broken down into the simple sugar units. Those units that are produced are called products. The products leave the active site. The active site remains unchanged, right? So the substrate has gone in. It is a very tight fit, and it's a perfect fit. That active site and that substrate are exactly right. The substrate breaks down into the products, into the individual parts, and those individual parts are then released from the active site. On the active site, the enzyme remains unchanged. So we'll look at the specific examples. So we have our carbohydrates. So we're going to start with our amylase enzyme. So we've got our amylase enzyme, and our amylase enzyme has a very specific site, active site. In this case, that the starch fits very neatly into. The hydrogen bonds, the bonds there that are represented by the blue line, they're broken down, and then the products, which in this case are the individual sugar molecules, they are released from the active site. And again, the amylase enzyme is then ready for the next set of carbohydrates that are ready to be broken down into individual sugar molecules. So for carbohydrates, you have your enzyme plus your substrate. So in this case, it is your enzyme plus your starch. And then your products in this are an individual sugar molecule. So if we then look at proteins, we have an enzyme and a substrate. So our enzyme in this case is called protease. And again, it has a very unique active site that will fit those different shapes of the amino acids very nicely. So the proteins fit into that active site, the bonds holding them together are broken down, and the amino acid, the products in this case, are released from the active site. You can see there, they're not bonded to each other anymore, they're single amino acid units, and the protease enzyme remains unchanged, and it is ready for the next set that is going to come in and is broken down into individual amino acids. So the final example we have here are the fats and the lipids. Now the enzyme in this case is called a lipase enzyme. So you've got your enzyme and your substrate. So you've got your lipase and your fat. The lipase then breaks down the bonds that are holding those fatty acids together, and you've got your products, which in this case are your glycerol and your fatty acid chains. Okay, so you've got your enzyme and substrate, your lipase and your fat. That then is broken down. They fit very nicely into the active site. The hydrogen, the bonds holding them together are broken down. Your products are your glycerol and your fatty acids, and they now are individual soluble units. On this slide is an activity for you to do. So using the knowledge that you've gained in the previous couple of slides, I want you to pause the video and I want you to copy and complete this table into your book. So you've got your substrate. Now you've got another one to know, which is when you've got carbohydrate, your enzyme is carbohydrase, and your product is a simple sugar or a glucose molecule. Now we've looked at starch, we've looked at protein, and we've looked at fat. So I want you to record in what the enzyme is called and what the product that was produced after the breakdown is called for that. So there are 6 sections there for you to fill in, hence the 6 marks. The section on inhibitors is in the higher content of your unit. So those of you who are going to be sitting the higher paper really need to pay attention to this one. Everybody should pay attention to it. Just because you think you're doing the foundation, don't have a look at this. So an inhibitor is a molecule that will go into the active site. It's similar in shape to a part of the active site, and it goes in, but it's not quite right, and it gets stuck, for want of a good word. So the inhibitor goes in and it blocks part of the active site. Now I've said before that in order for the substrate enzyme to form that complex, for them to stick together, there's a very tight bond between the enzyme and the substrate. And if there's something in there, then you're not going to get a very tight bond, and therefore it doesn't go in, it doesn't fit, so the whole reaction doesn't take place. So an inhibitor is a molecule that can fit into some of the active site of some of the enzymes. And whilst they occupy the active site of the enzyme, you can't break down the substrate molecules. So that leads to a lower rate of reaction. And rate of reaction is how fast you're taking your substrate and you're making your products for it. So an inhibitor is a molecule that attaches to the active site, blocks the substrate from being able to bind tightly into the active site. Therefore, the enzyme can't do its job. It can't break that larger molecule into the smaller molecules. So we have some questions now just to finish off this part 1 of this section. So just testing your knowledge, I want you to go back over your notes, go back over the video, look at it again, read it again, listen to it again. So question 1, why do food molecules need to be broken down? Right, there's 2 marks there. It's not, the answer isn't just to make them smaller, right? You've got to use specific jargon. You've got to use, we've talked about soluble, insoluble, absorb, all these different things. There's 2 marks there. You've got to give a decent answer. Question 2. What is the process of breaking down food into smaller soluble molecules called? 1 mark. That gives you a clue. They are just looking for a one-word answer there. Describe the 2 ways in which digestion occurs. Go back and have a look at the slides. There's 2 marks there. There's 2 ways. What is an enzyme? It's in red. Go and have a look. You should be able to give that answer off the top of your head. And then explain enzyme specificity. And again, there's 2 marks for that question. So again, they're looking for detailed answers with biological jargon in them. Active sites, for example, and so forth. Slide and the final question in the section that you have to do comes from March 2020, and I think it was the second question in the paper. It's question 6 here because there was 5 on the page before. So it says commercially prepared baby food often contains enzymes, and these enzymes start to digest the baby's food before it gets to the baby's stomach, and this helps the baby completely digest its food. So name the enzyme that would be added to commercially prepared baby food to help digest protein. Use your knowledge of enzymes to explain why a different enzyme would not digest protein. Look at the marks. The first one asks you to name an enzyme. There's 1 mark. They're looking for one word there. Second one, use your knowledge of enzymes to explain why a different enzyme would not digest protein, and they're looking for 2 marks there. So they're looking for a more substantial answer for that one. In part B, the diagram below represents part of a protein molecule. You should recognize that from examples I used earlier on it. Name this type of biological molecule representing the different shapes in the diagram that make up the protein molecule. Now they've told you it's a protein molecule. They've got the different shapes. What are they? Those individual units. Draw the products formed when this part of a protein molecule is completely digested. And there's 2 marks there. Now, I do want you to just be careful. I've seen this happen loads where people just draw random shapes. They have asked you to draw the products formed when this part of a protein molecule. They've given you a picture. It's very simple, and they've asked you to draw this part of the protein molecule. So draw this part of the protein molecule to gain those 2 marks there.